Okay, let's start Chapter 5 in Bio 101. So Chapter 5 deals with the plasma membrane, also known as the cell membrane. So something we already talked about with the plasma membrane, so there's a blow up of the plasma membrane right here. So something I've already talked about, it's the boundary of the cell. It surrounds all cells, right? And what it's going to do, it's going to regulate what enters and exits the cell. We'll get to how and why it does that, all right? So it is said to be a selectively permeable membrane in that it allows some materials in, but not everything in. It is composed of a phospholipid bilayer, so there's 2 layers of phospholipids, and it's embedded with proteins. All these purple things are proteins. Now, I do want to point out our plasma membrane is essentially colorless. The proteins don't have color of themselves. Okay? Now, the model that we use is what is known as a fluid mosaic model, in that the plasma membrane is essentially a thick liquid, like the consistency of olive oil, and it's mosaic. And a mosaic is made of smaller pieces. So you think about mosaics in art, they're small little tile pieces. All right, so let's take a look at the structure of the plasma membrane, because the structure of the plasma membrane is going to tell us how it becomes this selectively permeable membrane. So let's take a look at lipids in the plasma membrane. So the first are phospholipids. And so this is a computer generation of phospholipids, same here, okay? Now, we have 2 different parts of phospholipid. The upper part, so this part right here is known as the phosphate head, shown here, there's the phosphate head. And these parts here are called the fatty acid tails, so shown here, the fatty acid tails. Now, the phosphate head is said to be hydrophobic. So when we say something is hydrophobic, hydro means water, scratch that, it's hydrophilic. So hydrophilic, hydro means water, aphilia means loving. So the phosphate head here is hydrophilic, meaning it's water loving. So that means this is a molecule that is attracted to water, right? Or another way to say that is it's a polar molecule, okay? All right, next, down here, these parts, the fatty acid tails, have the completely opposite property in that they are hydrophobic. And a phobia is a fear. So these are molecules that are repelled by water, all right? So they don't like water. So these are non-polar molecules that are hydrophobic, all right? So what we have here is we have this molecule. One part likes water, the other part does not like water at all. So we take phospholipids and we place them into water. What's going to happen here is that the fatty acid tails are going to move away from water and towards each other, and the phosphate heads are going to orient towards the water. And so this is what you see, okay? So, this is showing a bunch of this here. And so what this does is it creates this interface between 2 different types of solutions. You can have 2 different types of solutions on either side of this. Because what we have in the middle, if we have this large hydrophobic area, so this large hydrophobic area, and what that's going to do is that's going to prevent any hydrophilic molecule, and that includes water, from being able to move through there. Okay? And this is a good thing. All right? So by preventing water from moving through here, if water could move through here, this layer could dissolve away, which would be bad, which would mean no one could take a shower because then you would dissolve away. All right? So it prevents water from going through here, so, and any other hydrophilic molecule from moving through there. So when we say the plasma membrane is a selectively permeable membrane, only small or hydrophobic molecules can move through. All right? Hydrophilic cannot, and that includes water. So what this also means then is that we can have a completely different solution on the outside of a cell than what's found on the inside of the cell. All right? So, what could be in our blood could be very different from what's in the cell. Or if you're a single-celled organism, what's in the water around you can be very different from what's inside your cell. All right, so let's take a look at other lipids that are found in the plasma membrane. So next is cholesterol. So you're seeing some cholesterol molecules here or there. So what cholesterol does is it helps stabilize the plasma membrane, helps keep it together. All right? Next are glycolipids. So you can see here, and there's another one back there. All right. Glyco means sweet, but it always refers to sugar. So a glycolipid is a phospholipid that has a carbohydrate attached to it. So we see that there and there and there. And these guys, and there's another one, these guys are used in cell recognition. And just hold that thought here. We'll talk about cell recognition in a second, because the next thing I'm going to talk about, are proteins that are found in the plasma membrane. So if we move a little further here to here, okay, so you can see that glycolipid, all right. So right here is a glycoprotein. So glycoproteins this is a protein that has a carbohydrate attached, all right. These are also used in cell recognition. So what do I mean by cell recognition? Cell recognition is, these are molecules that are used to help us identify cells that belong to us versus cells that don't belong to us. So, white blood cells. White blood cells are defensive cells. And what they do is they go around our body all the time and they bump into other cells. And if those other cells have the right glycolipids and glycoproteins on them, that white blood cell leaves them alone. If they have the wrong glycolipids and glycoproteins on them, it kills that cell. All right? So that's how we identify things that belong to us versus things that don't belong to us. All right? So, glycoprotein, a group of glycoproteins you might be familiar with are blood type. All right? So if you have A blood, you produce the A glycoprotein. If you have B blood, you produce the B glycoprotein. If you have AB blood, you produce both the A and the B glycoproteins. If you have O blood, you don't produce these glycoproteins. And so what do we know about O blood? Is that O blood is a universal donor. Everybody can accept O blood, all right? And the reason, this shows the loophole that's in our immune system, is that if you lack identification tags altogether, then our white blood cells don't do anything. If you have the right identification tags, our white blood cells leave you alone. If you have the wrong identification tags, our white blood cells kill you. Right? And if you don't have identification tags at all, the white blood cells leave you alone. And this allows us to put metals and plastics into us, unless you have an allergy. And an allergy is an overactive immune response. You're getting something into you that your white blood cells think is foreign and that wants to harm you, but in fact it doesn't. It's just misplaced in most cases. Okay, now, one of the problems with our phospholipid bilayer here is that it is a selective permeable membrane and only hydrophobic molecules can pass through. Because there's a lot of stuff that needs to get into our cells that we need in there that does dissolve in water, and that includes water, all right? So we need water and glucose and so on. So how do we get those things into our cells? It's through these things called transport proteins. So transport proteins, these are proteins that move substances across the plasma membrane. So the first one here is a channel protein. So this is a channel protein, and you can think of a channel protein just like you just stuck a straw through the plasma membrane. So these are proteins that act as holes in the plasma membranes or pores in the plasma membranes. And so if you are a molecule, the only restriction on you going through this is that you are small enough. So these guys are just size selective. And this is how water gets into our cells all the time, is through these channel proteins. Next is a carrier protein, and a carrier protein is going to move specific substances across the plasma membrane. So this is trying to show a carrier protein. Not all carrier proteins are going to require energy, ATP. Okay. Next are enzymes. Enzymes are often found along membranes, right? Enzymes increase the rate of chemical reactions without themselves being changed. Right? Often found along membranes so that we can have reactions occurring within the cell or outside of the cell. Okay. The last one I'm going to mention here, now this says signal transduction, but these are receptor proteins. So receptor proteins are going to bind to a specific molecule, and they're going to cause some intercellular response. All right. So, if you're giving a speech, right, you're going to secrete some adrenaline. Adrenaline is going to get your heart rate up, your breathing up, your blood pressure up. All right, this is all caused by adrenaline. One of the things you might notice while you're giving the speech is that your mouth goes dry, and that's because adrenaline inhibits the digestive tract, your digestive tract activities, and your salivary glands are part of your digestive system, so it's inhibiting them secreting saliva. That's why your mouth goes dry. Okay, now let's take a look at transport through the membrane. there are two major ways in which we move substances across membranes. One is through passive mechanisms. So passive mechanisms are without energy, and the other is active mechanisms, and those are with energy. So let's take a look at those passive mechanisms first. So the first one is probably a process you're familiar with already, and that's diffusion. Diffusion is a movement of substances through, from areas of high concentrations to areas of low concentrations. Okay, so here substances are going to move down their concentration gradient. They're going to go from high to low. All right, and how this occurs is that molecules are in constant motion. All right, so in a solid, in a liquid, and a gas. So in a solid, they're hardly moving. In a liquid, they're moving a lot. In a gas, they're moving crazily around all the time. All right, so what's going to happen there is those molecules are going to collide with each other all the time. All right. And so those collisions are what's going to spread these molecules apart. So we drop some food coloring into this bowl of water. The water molecules are going to bang into those food coloring molecules. It's just like the beginning of a game of pool. So you get all the balls into a rack, you hit into that rack, and it's going to spread the balls throughout the table, right? So that's microscopically what's occurring here. These water molecules are hitting those food coloring molecules, spreading them out. Well, as they get spread out here, they're getting hit by other water molecules, which spreads them out further, and so on and so on. And so eventually, you're going to reach equilibrium, or the concentrations are going to equal out. Meaning, if you took a sample here, or here, or here, or anywhere in there, you would find the same amount of red food coloring molecules to water molecules anywhere you took that. Now, when we look at this across membranes, we're going to move to equilibrium, right? So these molecules will eventually get to equilibrium here. If we're looking at two different solutions, or substances will reach equilibrium as well. All right. So what can diffuse through membranes? Well, I've already told you. Small or hydrophobic molecules can. So that's it. All right. So this is very important, though, in us getting gases into us and gases out. So we inhale oxygen into our lungs. And so the amount of oxygen in our lungs is a higher concentration that's found in our blood. So oxygen simply diffuses from the air in our lungs into our blood, and carbon dioxide moves in the opposite direction. So we inhale about 20% oxygen, 21% oxygen. We exhale out about 17%. We inhale less than 1% carbon dioxide. We exhale about 4% carbon dioxide. All right, let's look at a special case of, so this is showing it by our cells as well. So a special case of diffusion, this is called facilitated diffusion. So if you're facilitating somebody, you're helping them out. So in facilitated diffusion, this is diffusion of substances across a plasma membrane using a transport protein. And that transport protein could be a channel protein or a carrier protein. Now remember, carrier proteins are going to move specific substances. So this is how hydrophilic molecules get into our cells, and also larger molecules get into our cells. All right, so just an example here, after you eat, your blood glucose levels go up. On all the cells in our body, we have carrier proteins that allow the glucose into our body cells. But those cells have to be, or those proteins have to be activated by insulin. Insulin's basically like the key that allows those glucose molecules in. Without insulin, those glucose molecules don't make it into your cells. And glucose is our main molecule that we use for energy. Okay.